Good afternoon, everyone. This is Daniel Dibaba from Tennessee Clinical and Translational Science Institute. Welcome. Today, we'll discuss about factors to consider for a successful clinical trial. Can you all hear me?
Yeah,
All right, thank you.
thank you.
So at the end of today's presentation, you will be able to identify randomization methods. You will be able to understand masking and blinding in clinical trial and describe follow-up, adherence, and dropout, and identify analysis methods, including intention to treat and pair protocol. So in clinical trials, clinical studies, or clinical trials, study participants are randomly assigned to an experimental drug or biologic agent or to a similar looking agent called placebo. So what makes a clinical trial or experimental studies different from the observational studies is that in experimental studies or clinical trials, the investigator actively assigns intervention to the intervention group or to the intervention arm and compares the result or the effect of the intervention in the experimental arm to the effect in the control arm. So this is what makes an experimental study or clinical trial different from an observational study. In observational study, the investigator does not actively assign the participant into the exposure, into the treatment groups, but will observe what the participant have taken the intervention or exposure the participants have taken on their own interest or on their own volition. So intervention in clinical trial can also be a behavioral intervention or a procedure such as surgical intervention. So mainly the experimental study or clinical trials are trials where the treatment arm receives a kind of drug or a biologic agent, and then the effect of the experimental drug or biologic agent is compared to that of a placebo or to that of a control group. So an effective randomization is very important. It's one of the most important attributes of clinical trials. An effective randomization distributes confounding variables or characteristics evenly between the comparison groups. So one of the important attributes of clinical trials is randomization. We'll discuss this in detail in the next slides. So clinical trial is often called the gold standard study design, especially when the subjects are randomized to the intervention and the placebo or control arm. So the main reason is that potential confounding variables or variables or data that can define some of the differences between the compared groups would be uniformly or randomly distributed between the compared groups and create equivalent groups in clinical trial. This cannot be achieved in observational studies. Usually it's taken care of during the analysis, but in clinical trials, randomization uniformly distributes the characteristics of participants. For example, the age can be uniformly distributed in the compared groups. Gender can be uniformly distributed between the compared groups. Weight, heights, and many other attributes can be uniformly or evenly distributed between the compared groups. However, association in clinical trials are not proof, as otherwise we do not have to do multiple clinical trials. Even though clinical trials are assumed to be the gold standard and the best source of evidence, scientific evidence, associations from clinical trials cannot always be taken as kind of causal, because if that were the case, we do not need to do multiple studies on the same topic. So when we look at the treatment arm in clinical trials, the clinical trial arms are the intervention arm and the control arm. The intervention arm receives the intervention under study. So this can be a drug or a medicine. It can be a biological agent. It can be, for example, when the patients are treated, or the clinical trial is about using different kind of material for hip replacement or knee replacement, or for treatment of femoral head fracture. It could be, one can be metal, another can be ceramic or something like that. And then it's compared to a similar looking material or drug, which is what we call placebo. So placebos are inert treatment is intended to have no biological effect other than the psychological benefit of receiving treatment, which itself can have a powerful effect. Sometimes this kind of effect, if the placebo has some effect, the patient feels they are receiving the treatment, and sometimes can tell like they are receiving some benefit from the placebo. It's called a placebo effect. So as I said, randomization is one of the very important quality of clinical trials. In the 20th century, Ronald Fisher, the father of statistics, developed a method called randomization that accounted for confounding variables or external source of differences in compared groups. Each subject is assigned to the treatment arms using a random assignment mechanism. We'll look at in the next slides some of the specific randomization methods. So randomization reduces bias due to confounding variables. The confounding variables are characteristics of the participant that can be different between the two groups, the intervention group and the control or the placebo group, that can result in differences in the outcome. For example, a drug might be given in clinical trial for reducing or for reducing cholesterol levels. If we don't consider the two, if the two groups are not comparable in terms of age, gender, BMI, and many other characteristics, the difference might be due to the difference at the beginning of the study, no rather than the drug being compared. So randomization reduces such kind of other source of bias or confounders. And when we say randomization, it means a random assignment of a participant into the treatment arm and the placebo or control arm. So randomization evenly distributes confounders between the compared groups. So if you have any question while we go, you might type in the chat or raise your hand, and hopefully I'll be able to see you. So when the intervention and placebo arms are compared for the average distribution of the participant characteristics, if the randomization is achieved as planned, there must not be significant difference between the compared groups, except by chance alone. And usually that should be very, the difference due to chance should be very small. And if randomization has been achieved, that there has not to be differences in the participant characteristics between the intervention arm and the control arm. Otherwise, sometime I remember one study that was published and it was reviewed by some professors from Indiana University. The study was published and it was published as a randomized clinical trial, but there was significant differences between the intervention arm and a control or the placebo arm. And so the reviewers, like after the study was published, um, they wrote to the editors that randomization actually has not been achieved because there was a significant difference. Eventually, the study was pulled back, it was redacted from publication. So when we compare the intervention arm and the placebo arm, we have to make sure that there are no significant differences in participant characteristics between the two compared groups. If we are going to say there is randomization that has taken place for the assignment of participant. So but the validity of trial depends strongly on the extent to which the random assignment to treatment arm is the only determinant of treatment received. So this is what we call internal validity. So internal validity of study is when the study has been properly conducted and there is no selection bias or any other source of bias. And the association can be taken as solely due to the differences in the effect of the intervention and the control, the comparison group. If we can attribute the differences only to the treatment, and then we can say the study has internal validity. So the validity of trial depends strongly on the extent to which the random assignment to treatment arm is the only determinant of treatment received. So besides randomization, validity also depends on other factors such as the study duration. If the clinical trial is of short duration, sometimes we might fail to see effect over a long duration of time, or we might see some differences that is only short-lived, and our conclusion might be biased as well. So validity of clinical trial might also depend on the duration of the study. It can also depend on the sample size. As you all know, if the sample size, the number of participants in the study is very small, we might fail to see small differences that are clinically relevant. But if the sample size is very large, and then we might even be able to see small differences because the statistical power is directly related to the sample size or the number of participants. Training of the study personnel might also be one of the factors that might affect validity, and the other is masking. We'll discuss about masking in the next slides. So the average effect of the variation in confounders or participant characteristics should be less than that of the experimental agent. So when we say there has not to be significant differences, except that might occur by chance alone, we mean that the average effect of the variation in confounders or participant characteristics should be less than that of the average effect of the experimental agent or intervention. So does the randomization balances covariates or variables, uh, additional variables, predictor variables between the treatment arms? When we say covariates, these are the factors that can vary between the compared groups, such as age of the participant, race of the participant, it could be gender, it could be BMI, it could be many other factors, metabolic differences and the likes. So those are covariates. They vary between the two groups, and if we don't balance, that means, for example, if we have 30% of participants who are below 30 years of age in one arm, we have to have about the same percentage in the control arm as well. So that means we are balanced. There has been balance in the distribution of the characteristics, the third characteristics between the treatment arms. Another one, randomization avoids or reduces selection bias that may be introduced by staff or the investigators. So when we say selection bias, this means if the selection of a next participant when assigning treatment or a placebo depends on the assignment of the previous participant, we say there is a selection bias. So if after assigning one participant, the next participant's assignment into treatment or a control arm depends on the previous one, we say there is a selection bias. So, but randomization kind of reduces that risk of selection bias. Randomization also improves internal validity of the study. And by internal validity, we say the absence of bias and confounding and all sorts of factors that might distort the relationship between the treatment and the outcome of the intervention, the outcome of the clinical trial. So a study has to have internal validity to so that the conclusion can be accepted by the scientific community. So there are three different randomizations. One is a simple randomization. This is a simple randomization. When we say simple randomization, this is like the coin toss or selecting participant from a pool of participants just by randomly picking participant from the whole population. A coin toss is one. So if when we throw a coin, toss a coin, if head comes up, we assign the participant into the intervention group, and if tail comes to the control group, just like that. The problem with that is as such randomization is not replicable. We cannot repeat this after, say, if another team of scientists want to reproduce our result, and they want to start by allocating participant into the active arm or treatment arm into the control arm. If simple randomization such as coin toss is involved, the result cannot be replicated. So that's one of the issues. Block randomization is another randomization mechanism. In the block randomization, the experimental divides the participant into subgroups called blocks, such that the variability within block is less than between the blocks. So randomization can take place within the block. So if, say, we have 24 participants, we can create 6 blocks each with a block having 4 participants. And within the four participants, they all have similar characteristics. And all those participants are randomly assigned into the treatment arm or into the control arm. So a block randomization is one of the randomization methods. It can be replicated, and there are statistical methods or softwares that can help in the assignment of participant into the treatment arm and into the control arm. The third one is a stratified randomization. So in the stratified randomization, we divide the participant by certain attributes and randomize them to treatment arm. So for example, we can classify participant by gender, for example, into male and female. And we randomize among female, we randomize them to a treatment and a control arm. So there is the female participant receive both the treatment or the control, and the male participant also are randomized. So the effect of gender can be taken care of like that by stratifying into by gender. Another example is randomizing based on whether the participant is smoke or not. So once we divide them into smokers and non-smokers, we randomize the smokers into the different treatment arm, and non-smokers into the different treatment arm. During the analysis, we can keep this stratification so that we will only compare the participant among smokers and also participant among non-smokers, and then finally we can pull the result on the effect of the intervention. So the intervention actually can be a drug, it can be surgical treatment or something else, or it can be behavioral. We can use simple or block randomization as a method after we conduct a stratified randomization. Another common form of stratified randomization is when we we list the whole participant, and then we take, for example, we want to select 100 participants out of 1,000 eligible participant, and then to get the 100 participant, we start by dividing. So we divide 1,000 by 100, and then we get 10. So we select every 10th participant and include in our study. And that's one of the stratified random sampling. But the stratified randomization is when we divide into two groups by a given attribute. Oops. So we said among the mechanisms of randomization, a simple coin tossing is one method, especially for the simple randomization. And the problem with this is that it's not replicable, and thus it's less reliable, and we don't commonly use this one. And the other one is using software. So there are a couple of R packages that help randomize, that helps to create a sequence of participants that can be assigned to the treatment arm and to the placebo arm. One of such R packages is randomized R. This helps us to calculate um, the amount of bias and the amount of bias due to selection bias or chronological bias, and the final statistical power that we can achieve based on the randomization that was conducted. So randomized R is one of good statistical package out there that can help us generate the list of or sequence of participants that can take that can be assigned to the intervention arm, and that can be assigned to the control arm or placebo arm. And another one is randomized in the experiment R package, and block random function in the psych R package. So there are a couple of R statistical packages that can help us create the sequence of participants that can be assigned to the treatment arm and control arm. So we can accomplish randomization like coin toss, but the results are replicable using seeds, what is called seeds. So the seeds enables are gives the same result again and again. So for example, we can produce the same results as what we did today by providing some kind of seed numbers, which is a list of 4 or above numbers that kind of captures the state of the are packaged at the moment. And so the result can be the same every time we use the same seed. So we can use seeds to achieve replication of the result. So somebody else can use our data and get the same kind of assignment again and again. And so that's one way to achieve the replication of the result. We can test for selection bias and, like I said, the chronological bias and power loss due to imbalance. So when we say chronological bias, this is a bias that can happen, or differences that can happen due to the effect of time on participant assignment to treatment arm. For example, if clinical trial starts this year and does not finalize, and it extends over several years, there might be some changes. Treatment might change. Some might be some improvement might come into the treatment, and or many other factors can change. The time period can have effect sometime. For example, um, the the metabolism of individuals might change during seasons. So it might be different during winter and summer and the likes. So a time might make an impact, and those kind of things are considered chronological bias. So it's a bias due to time effect on the study assignment and the effect of the treatment on the outcome. So we can test imbalance. So like I said, if there is imbalance in characteristics or covariates, that means the randomization was not as expected. And we cannot completely say there was randomization. We cannot say there was an effective randomization in such situations. So masking or blinding is another attribute, very important attribute of clinical trial. Um, so participants are not told which treatment they are assigned when they are recruited and randomized to receive either the experimental agent or placebo, or there would be a control group. If they are a control group, of course, we cannot say there is masking or blinding, because the control group know that they are not receiving the treatment under investigation. But in other situations, like when there is a placebo arm or a control arm that receive another treatment, um, we can blind or mask the participant. So everybody thinks that they are receiving the treatment under investigation, and the investigators are not informed who is receiving the treatment and who is receiving the placebo or a comparison arm. So in this situation, we call, when the investigators are masked and the participants are masked, we say there is a double blinding, which refers to the participant and investigators being unaware of what treatment arm the participant is on. So sometimes a person who assigned the participant into the two groups, into the treatment and the control arm or placebo arm, is not involved in statistical analysis or interpretation of the result. And uh, so until at least after the study has been completed. And so masking is one important attribute, so that, for example, if participants know what they are receiving, one, it might affect their participation. They might drop out of the study. They can also have some kind of, they can make up their mind, and especially when the effect is some kind of subjective, the outcome might be kind of biased. And the same can be said about the investigators. If they know what the participants are receiving, so which arm the participant belong to, they can also make a kind of biased assessment about the outcome of the intervention. So masking or blinding takes care of that and reduces the effects of bias introduced by the investigators or the patients or participants. So the purpose of masking or blinding is to reduce selection and chronological bias and to reduce perceptions such as placebo effect affecting the outcome. A third clinical trial characteristics that usually are required to be well described is the follow-up and dropout rates. So when we start the study, maybe 100 participants are randomly assigned to the clinical trial or investigation arm and the control arm. So say 50-50 would be assigned. And at the end of the study, if there is no some incentives to increase incentives to increase compliance and participation, that some participant might drop out of the study. And this can affect the conclusion and also can lower the statistical power that our study achieves. So incentives to participants such as fuel rewards, bus ticket, limited financial bonus might enhance participation. The level of dropout rate must be described and as one of the clinical trial quality metrics. So along with the dropout rate, the analytic metric should also be described. So another attribute that we need to describe in clinical trials is, especially after the study has been conducted, we have to describe about the adherence. So adherence is the degree to which the participants comply with the study instructions on how to take agents, dosage, frequency, visit to trial centers, and behavioral habits. So adherence to the study agent can be checked by asking the participant directly and asking the subject to return the remaining medication at the end of the study, or by counting the remaining medication in the middle of the study, or using biochemical measurements. So adherence is very important. Like participants are assigned to the experimental groups, and at the end of the trial, maybe some of them have not taken the medication, and then that means even if we might do the analysis on all the participants who have been assigned at the beginning, there is some kind of bias because the effect we see are not exactly what has been and the fact that they have not taken the medication properly might have an impact on the result of the study as well. So when we come to analysis strategies, adherence and the dropout rate and follow-up are very important, and they can affect the analysis strategies that we choose. There are at least two clinical trial analysis strategies. One is the intention to treat analysis. So in the intention to treat analysis, the data on all randomized participants are analyzed. That means even those who have, those who do not adhere to way the intervention and the control arm has to be taken, they are included in the analysis. And those who have also quit or are lost to follow up, they are also included in their data, at least their baseline data is also included in the analysis. So all participants are included in the analysis regardless of compliance to the protocol. I don't like this, but some people say this is a preferred a strategy for analysis of clinical trial. The pair protocol analysis is the other one, and this analysis only data on participants that adhere to the study protocol are analyzed. And those who have properly taken the assigned treatments, including the dosage, frequency, and the likes, and the time, and all that. So when Data only on those kind of participants is analyzed, and conclusions are based on those kind of data. We say the study followed the pair protocol analysis strategy. So when we talk about clinical trials, especially in the case of drug clinical trials, there are two different different phases. One is the FDA clinical trial phase, and the other one is NIH clinical trial phase. So, and today we'll look at the FDA clinical trial phases. So the FDA clinical trial phase starts with phase 0. In this phase, the drug under development is first tested in few humans. Usually the sample size is less than 10 participants in small doses to test drug safety. So the first phase is the phase 0, and the purpose is to test whether the drug is safe in human population. Usually the preclinical phase of clinical trial takes place in animals like mice and the likes. And once the drug is observed to have some kind of beneficial effect in treating certain disease, the next stage is to conduct the clinical trial in human population or in human participant. So the first phase is phase 0, and the purpose of the clinical trial is to look at or to examine the safety of the drug in human population. So in very small doses. The next phase is phase 1, and this test is the best dose of the new drug, the best dose of the new drug with few side effects in about 15 to 30 patients. So this is also conducted in a very small number of patients. And the purpose of the clinical trial is again to test the safety of the drugs in human population. And in phase 2 clinical trial, it also further assesses drug safety and efficacy. So when we say efficacy, this means the effect of the drug in achieving the desired outcome. So again, a small number of participants are usually tested, like below 100. And the third phase is this compares the new drug to standard of care if one exists. And usually this is conducted in about 100 or more patients before the drug is approved by FDA. And the last phase is um, a clinical trial on drugs that have already been approved by FDA for a short, long-lasting side effects. So the difference between the FDA and NIH clinical trial phases is that in the NIH clinical trial, we do not have the 0 phase, but the started with phase 1, and also even though the purposes are similar, the sample size are also different. So the last one is where after FDA has approved a given drug, the trial is conducted in 1,000 or more participants. The third one is when the trial is conducted in a few hundred participants. So the differences are the sample sizes are different, and it doesn't have phase 0. So overall, the metrics of clinical trial that should be described in method section in very detail are about randomization, what kind of randomization mechanisms have been conducted, and whether randomization has been achieved. This has to be described, and we have to describe what method we use. Is that a simple randomization where we just toss coins? Did we use random numbers to assign participant to the intervention arm and to the placebo arm or control arm? Did we use some statistical software to make the assignment of participant into the different arms, which mostly ensures the randomization? So we have to describe this in detail, and a peer reviewer will look at whether the randomization that has been whether randomization has taken place in the clinical trial, and whether the randomization was well described, and whether the randomization was actually effective, whether it was as described and can be taken as randomization has been conducted. So the quality of randomization will also be assessed. And it's one of the quality metrics of clinical trial. And the other one is blinding or masking. And blinding or masking means the investigator or the participant do not know the intervention drug and the placebo drug, or what arm they are on. So everybody assumes they are on the treatment arm, um, and they cannot differentiate whether they are on the treatment arm or on the protocol, on the control arm. So this can reduce bias that can be introduced by the investigator as well as by the participant. And another metrics of clinical trial is having appropriate comparison group or a placebo. This has to be described as well in very detail. And the other one is describing the dropout rates. So uh, what was the follow-up, what are the follow-up of the study look like, and how many participants dropped out of the study, and how was the analysis conducted? What was the how do we justify the analysis method that was elected, whether it is intention to treat analysis or per protocol? So when we also talk about the different analysis methods based on the study outcome, with if it is a continuous variable, we can take like a t-test or something, analysis of variance and the likes, or multiple regression. And if the outcome is a binary outcome, or an outcome that has a yes or no type values, and then we can use the logistic regression analysis. Or if the study was conducted over an extended period of time, and we can conduct kind of survival in terms of waiting for certain outcome, we can also use the survival analysis. So the specific analysis method is different based on the type of outcome and the duration of the study and the likes. But the strategy can be whether to include all participants, regardless of whether they are compliant in terms of an instruction, or and doing the analysis only among those who were compliant. So generally, that was all. Thank you very much for your attendance. So now we have time, and if you have any question.
Right, thank you, Daniel.
Thank you, Rongshun. Does anybody have a question?
You mentioned the phase 1 is for next things. So, what's the phase for test for animal before test for person?
Oh, the phase for animals, and actually for animals, we don't have this kind of like the 4 different phases.
Okay.
Yeah, so they are.
Phase 0 is a test for animal or directly test for human beings?
Pardon? Can I say that again?
I mean, phase 0.
Oh,
Yeah.
it's in humans.
It's less than 10%, right?
Yeah.
So we don't need to test for animal.
Yeah. So almost all drugs are first tested in animals.
Okay.
And when the investigators see that drugs have beneficial effect in animals, and then the next step is the clinical trial. And the phase B for that is called preclinical.
Okay. Thank you.
And the preclinical tests are in animal models.
Okay. Thank you.
Yeah. So thank you very much. To look at the services the Tennessee Clinical and Translational Science Institute provides, please go to our website. You can see the services that we can provide, among which are bird clinic is one of the main services that we provide. In bird clinic, we assist clinical investigators and faculties in study design, in epidemiology, biostatistics, and research design. And so that means we help them in determining the number of participants that they need for their research. And we also help them with the way to collect the data, the participant data, and resources for recruitment of participant. We also help them with what kind of data they have to collect for their study and how to analyze. So we'll help them with the statistical analysis plan and statistical data analysis as well. So we provide those services, and we are planning to also provide manuscript review, scientific review. In the future, we are also planning to start scientific review of clinical trials. Very soon, we'll be starting to provide that service as well. So thank you very much. If you have any question, we have a lot of time left, so we can discuss. If you have questions, you can also email me at my email. So if anybody person has a question, this is a good time to ask.
Bye, Daniel. Yeah, take
All
care.
right.
How's
Have a
the
great
snow
day.
there? How many inches there?
It's about 5 to 7, 8 maybe. It's super cold.
Okay, yeah.
Yeah, today it was relatively better, but yeah, let me stop.
When it is snow, even